With that, I'm going to turn it over to our very first presenter from Florida Gulf Coast University. Bass Dye, are you on the line?
I am. Can you hear me?
We can hear you now. Take it away. Thank you.
Okay, great. Thanks. Well, thank you for letting me present our research today on oyster larval transport and modeling within the Charlotte Harbor system. This work was a collaboration of various people from FGCU, SCCF, and the DHI Modeling Company, as well as being funded by the FGCU Whitaker Center Blair Summer Research Scholarship. Next slide, please. So it's important to study oysters because they provide various ecosystem services, such as filtering the water column, providing habitat for various organisms, as well as many other services. However, it's quite challenging to study larvae as well as larval development and transport because of the scale. They are these small organisms located in large and complex environments. And you also must account for different biological processes such as their development as well as tolerance to environmental conditions, but also account for how these larvae are transported due to the physics of their environments, as well as the interaction of these two processes. However, despite these challenges, just an understanding of basic questions such as where larvae come from and where larvae go can provide very valuable information for the management and restoration of oysters. Next slide, please. Our study addressed these challenges in 3 steps. The first being developing a hydrodynamic model to simulate the physical processes of the system. Second, by developing an ecological template composed of various biological parameters of the eastern oyster. And then third, by coupling the hydrodynamic model with this Ecolab template to simulate oyster larval transport within the system. Next slide, please. So our study was developed with 3 research objectives in mind. First being, are we able to accurately model oyster larval transport within the system when compared with field-based measurements? Second, if successful, can we then use this information to determine larval connectivity between different field-based oyster reefs? And then finally, using all this information, are we able to inform oyster reef restoration and management? Next slide, please. So our study began by first developing a hydrodynamic model. I think it's important to point out that this certainly wasn't the first and likely won't be the last hydrodynamic model developed for the region. However, our model in particular is a 2D hydrodynamic model covering the entire Charlotte Harbor system. We chose to use the Mike software from DHI because it allowed for the easy coupling with the Ecolab template for the ultimate simulation of the larval transport. Some characteristics of our model include as a fine resolution of around 200 meters within the estuary. It is forced with realistic winds, tides, as well as freshwater flows, and also incorporates salinity data from various recon monitoring stations. We chose to use a 2D model mainly to reduce computational time, as well as considering the shallow nature of the bay. The model does quite a good job recreating the dynamics of the system. And if you're more interested in kind of the model development and calibration in a circulation dynamics study, we published a paper in 2019. Next slide, please. So the next step of our study was the development of the Ecolab template, which is a customizable ecosystem modeling tool, which we designed specifically for the Chrysostera virginica oyster larvae. The template includes parameters such as a larval development period, which after the agents, which I'll refer to later in the study, the agents are the simulated larvae. So once these agents are released within the system or in the simulation, there's a 10-day period where they're unable to settle on suitable oyster reef substrate. There's also differing settling velocities associated with the agents throughout the simulation. So during that 10-day development period, the agents are neutrally buoyant, and then after that 10-day period, they are given a small settling velocity to help recreate different settling dynamics. Also, natural mortality in the form of a type 3 survivorship curve, where there's high mortality early in life and less mortality later in life, is incorporated into the template. So once the agents are released within simulations, the agents are randomly subjected to this natural mortality. And if selected randomly, then they are subsequently removed from the simulation. Next slide, please. Some additional parameters include agent sensitivity to differing salinity ranges. Those ranges are shown in the figure. But for example, an agent can survive for at least 6 hours in water less than 5 PSU. If that agent is not transported to a region with higher salinity, then that agent is classified as dead and removed for the simulation. And the similar circumstances occur for those other thresholds shown in the figure. And also, for an agent within the simulation to successfully settle onto oyster reef habitat, it must meet 4 different requirements. First being, it must gain competency, meaning that it successfully developed through that 10-day period. The agent must be located within 5 meters of 4 oyster reef monitoring sites, which I'll show in the next slide. And the current velocities around those 4 sites must be less than 1 meter per second. Next slide, please. So in order to validate our larval transport simulations, we utilized oyster spat settlement and gonad condition data collected during a long-term study performed by Dr. Swani Vleddy and various other people from FGCU. The monthly sampling took place at the 4 sites shown in that larger figure, with those sites representing the salinity gradient within San Carlos Bay. And site 1 was also the existing extent of oysters at the time. So the gonad index provided us when and from which sites to release the agents within the simulations. And then the spat settlement, or the settlement data, was used to validate our agent settlement at the conclusion of our simulations. Next slide, please. So we conducted 3 separate simulations during the months of July, August, and September 2011. For each of these simulations, 2,500 agents were released from each site, which was indicated by the gonad index field data to be spawning during that model simulation time period. Since we didn't know the exact timing of larval release or on what tidal cycle they were being released, we released the agents during an ebbing tide because this would provide kind of the worst case scenario for larval retention within the estuary. And then finally, the simulations were conducted for a total of 25 days, which is typical of a larval life cycle. Next slide, please. So unfortunately, we're unable to directly compare the measured and simulated settlement because the simulated settlement does not account for any post-larval settlement mortality. However, what we can do using these 2 data sets is to observe the trends between the 2. It's important to note that the data presented in both these figures is in terms of percent settlement, which is calculated by the number of either the measured spat or modeled spats settled at each site, and then those values were divided by the total amount settled. So if we first look at this left figure, we have percent settlement on, or it's important to note that the data shown in this is actually a combination of those 3 simulations. So combined settlement, measured and simulated, because this kind of gives us a better idea of how well the model performs overall as opposed to within the individual simulations. So we have percent settlement on the y-axis. We have the different sites on the x-axis. Measured settlement is in gray, the simulated is in black. And what we can see is that site 1, there's a pretty good match between the 2 data types. As we move to sites 2 and 3, the percent settlement is within about 10% of each other. And although the simulated settlement is quite less at site 4, we do see a similar trend between the two data sets in which we have increasing settlement moving from site 1 to site 3, and then that settlement is reduced moving from site 3 to 4. If we look at this right figure, we can then look at how well each individual simulation compared. So once again, we have percent settlement on the y-axis, we have the individual simulations on the x-axis, and those simulations are broken up by that vertical dotted line. And we once again see there's a pretty good match between the percent settlement of the measured and simulated data. Site 4 is obviously performing less well, but we still see that increasing trend of settlement from moving from site 1 to 3, and then that reduction in settlement moving from site 3 to 4. Next slide, please. So the model's able to reasonably recreate the measured settlement patterns when compared with the measured data. So that allowed us to look at connectivity patterns between these 4 sites. So the colors in these various figures indicate from the release site of the agent, the release location of the agents would settle at each site. So we have the blue agents released from site 1 in blue, agents released from site 2 in gray, and site 3 in yellow. So if we first look at the top left figure, this is our July simulation. Agents were released in sites 1 from sites 1 and 3. And we see at site 1, there's at least 60% of the settlement were actually from agents released from that site. So this is exhibiting self-recruitment, as well as we move to the downstream sites, we see at least 60% of the settlement is from agents released from site 1. And we see a similar result in the top right figure, where at least 60% of the settlement were from agents released from that uppermost site 1. If we look at the bottom figure, which is our September simulation, agents this time were released from sites 1, 2, and 3. And we once again see that at least 50% of the settlement were agents released from site 1. So these results indicate site 1's role as an important larval source to the downstream sites, but also it shows the roles that the individual sites play with supplying agents to each other's sites. Next slide, please. Interesting patterns also emerge when we look at the locations of the agents at the end of each simulation. So there are 4 quite distinct patches of agents that occurred in each of the simulations. We considered the groups 1 and 4 to be lost agents, because these were agents that were transported out into the Gulf and generally did not return. And agents shown in the circles 2 and 3, we considered as a surplus, because these are agents that were retained within the estuary. However, they did not ultimately settle. So these results indicate that under the 2011 hydrological conditions, which had on average less than 80 cubic meters per second of freshwater inputs from the Caloosahatchee River, that a sizable portion of the larvae are retained within the system. Next slide, please. So some important research outcomes. We were able to establish a novel oyster larval transport model for this system. And using those model outputs, we're able to determine connectivity between 4 of the reefs. And the results are important because they provide supporting evidence to Dr. Valletti's previous findings of site 1's role as an important larval source to those downstream sites. And also there's a sizable larval surplus at the end of the simulation, which could provide future insight into potential oyster reef restoration sites. As with any good modeling study, there's certainly caveats and limitations which need to be addressed. The first being that this current study is only validated for the low to moderate freshwater flow inflows. There's definitely a possibility that there's some uncertainty in in the parameters we chose in the Ecolab template, although they were taken from the literature. And currently, within the simulations, the agents were only allowed to settle on those 4 measured reefs. So it's certainly possible that those surplus or lost agents would be reduced if the agents were allowed to settle on more reefs within the area. Next slide, please. But the important thing is was initially a pilot study, and this work is ongoing. So our next step is to couple the Ecolab template with a 3D model. We are also conducting a field campaign in the coming summer months, which will expand those settlement sites, as well as collecting larval concentration, which will give us an indication of larval supply within the system. We're working to continue model sensitivity testing, as well as we're currently writing up these results for publication. So we'd appreciate any comments or suggestions you all may have. Next slide, please. And with that, I thank you, and I'll try to address any questions you may have.
Great. Thank you so much, Bass. We really appreciated that. And I think this just highlights how important students are in our community for assisting our natural resource protection efforts. So right now, for all of you online, either through Zoom or YouTube, if you wouldn't mind going to menti.com, menti.com, and using the code listed at the top of the screen, 639808, you're going to be entered in where you'll see this question: submit questions for student research on oyster larvae within the Shala Harbor and Caloosahatchee Estuary. So if you wouldn't mind going ahead and entering any questions you have right now in Menti, then our presenter will start to see those and be able to answer those for you.
So the first question is, will this data be made publicly available? Yeah, absolutely. I'd be happy to share the presentation and the kind of the draft manuscript we have, or any other information people would be interested in. When you extend this project, do you plan to include Estero Bay as either a source or a recipient of larvae? Yeah, we definitely could. We didn't have any field-based measurements there, so if someone could provide us with those measurements, we could easily include that in the simulations, and that would actually be great to include.
All right, we still have a few more minutes. So for those of you online, feel free to continue to enter your questions.
So the next question is, how many larvae have been released? If this is within the simulation, there was 2,500 per site. So for those first two simulations, there's 5,000 released. And then for the September, there's 7,500 released. And we actually did sensitivity testing to try to determine if the amount of larvae we released in the simulations changed our results. And we tested everything from 2,500 up to releasing 10,000 from each site. And it did not change the results. It just had a greater proportion of settlement. However, that same proportion of settlement was the same regardless of how many we released. So that's why we decided to release 2,500, because it reduced computational time while still giving us consistent results. Have you looked at species of seagrass and larvae? So I've only worked on oysters, but this particular template could easily be adapted for other organisms. So that's kind of the utility of this template. As long as you know kind of what parameters are needed, you can essentially just plug those values or equations into the template. So if you're interested in seagrass or other organisms, you could definitely be able to develop a template for those specific organisms. Are there declines in oysters in Caloosahatchee? So I don't think I'm the best person to answer that, because I have been living on the West Coast for the last 2 years. But I'm sure Eric Milbrand or someone better qualified could answer that. I apologize.
Oh, great. With that, we're going to wrap up this session.